Hello, I'm Neil Spooner, the Chair of the Patient Sampling Interest Group, and I'd like to welcome you to our latest webinar, Output from the PCSIG Clinical Trial, Impact of Home Blood Sampling on Recruitment and Retention. Really excited to bring you this webinar. It's been a long time in the making, as you'll see as we present to you. Your presenters today are Robyn Rourick from Genentech, Mike Walker from Walker Bioscience, and I have the privilege of also speaking today. So in my role as chair of the Patient-Centric Sampling Interest Group. Before we get started, just like to say some quick thank yous to all the sponsors of Patient-Centric Sampling Interest Group, without whom we wouldn't be able to run our activities and these webinars every month. If any of you have ideas for webinars, please write to us, let us know. At the moment, we are booked out until June 2026. But we're always looking for new ideas for webinars to do with patient-centric sampling. So please let us know if you know anything. Just note that there should be plenty of time for questions at the end of the webinar. Please submit these via the chat function, which is on the right-hand side, and we'll read those out at the end. The webinar is being recorded and will be available hopefully next week through our YouTube channel to view again. And it will always be free to view through our website and our YouTube channel. So with no further ado, I'd like to hand over to Robyn Rourick, and I'll speak to you later.
Thank you so much, Neil, and greetings, everyone. It's a pleasure to be here today to discuss our study with you. So I'm going to start by focusing on the design of the alcohol consumption study. And the intent of that was to evaluate the impact of home-based blood samples, the collection process on participant recruitment and retention compared to our traditional and clinical methods. And you can see the device which we utilized, which was a Tazo MS-20 blood collection device on the upper arm of the individual noted on the slide. And to begin, we focused our advertisement and recruitment in the Seattle metro area with the intention of trying to make it feasible for participants to partake in either of the arms, so either the in-home or the in-clinic study arm, because our goal was to have our results driven by the perception and the experience with the utilization of the device, as opposed to any issue with such as long driving distances, so on and so forth. So the volunteers that were identified were randomized across two groups. So either the enrollment in the at-home group or in the clinic group. And as you can see in the middle section, each of those were inclusive of 3 sampling events or sessions, if you will. So in the at-home, when an individual was registered, a kit was sent to the home, and then once that sample was received back, the subsequent kit was then provided. And similarly, in the clinic, there were an initial appointment that was made. The sampling was done in clinic, and then a subsequent appointment was made for the following sampling. And then the duration of that sample sessioning was about 1 month time frame. And then following the completion of each of the 3 samples collected, there was a questionnaire that was provided to each of the participants. From then, we moved on to data analysis, which involved looking at the portion consenting, the returning of the samples, the time to consent, the time to return, and then data that was collected through the questionnaire. And following my discussions, Mike Walker will talk us through more specifically with regard to analysis. And then we moved into sample analysis using a validated method to determine the amount of phosphatidylethanol, or PEth, which is a biomarker that indicates recent alcohol consumption. And then the results were ultimately shared back to the patients as well as the site. So moving on. This is a high-level study timeline, and it kind of gives you a sense of the duration from the initiation and the planning to the actual last subject in. And it was a fair amount of time. It was a 4+ year timeline, and it certainly involved a lot of challenges and various points where we had to navigate, particularly COVID, as you can see, stood out. So we initiated our thinking through the PCSIG working group in early 2019, and we were leveraging insight that had been gained through the alcohol consumption study, or dry January study, if you will, through Christophe Stove's lab in Belgium. And our initial intention was to take advantage of dry January, where, you know, people after the holidays, they come back enthusiastic. It's a new year. I'm going to cut back on my drinking. And we thought that that could be a great attention getter for folks to want to enroll. But then shortly after that, we were hit by COVID, and we had to navigate that space, you know, quite quickly. And then that was also compounded by a transition in a study investigator, which required us to do some further onboarding, another individual, and to align with strategy, so on and so forth. So at one point through the course of COVID, the PCSIG working group made a decision to put the study on hold. And it was about 2021 or so where we started to rethink our strategy and consider opportunities for advertisement using social media. And we did a number of ads on Facebook and began to get some uptake. And our first subject was actually enrolled in August of 2022. And, you know, as we paid attention to the enrollment, it became very clear that we had to think a little bit more strategically, ways that we could appeal to students and various social groups, and began advertising in various sectors. And then also moved into a stipend where we began to offer a $10 fee for each of the 3 samplings that you noted on the previous slide, and then also some ad revisions. We started to pay attention to the demographics. We saw more of a steady increase with females versus males, so we did some ad adjustment, actually in the colors, believe it or not, for our Facebook advertisements. As we enhanced the colors and moved more towards blues, we actually saw an uptick in males, which was interesting. So our last subject in was July of 2024. So pretty much a 2-year time frame to receive the optimum number of recruiters. So moving on. So in terms of sample collection, both groups completed the informed consent. And the same sampling device, again, the Tazo MS-20 was used for both the in-clinic and the at-home groups. Collected samples were stored at 4 degrees C until the analysis of the PEth biomarker. They were inspected for sample collection quality, both at the clinical site and the analytical lab, and we'll share some details about that a little bit later. And the in-group specifically had appointments made for the clinic, and then the samples were collected by the clinical staff. The at-home group, their first sample kit was sent to the address upon confirmation of their participation, and then instructions for the use of the kit were included in the box. And then also they had opportunity to access support through a customer service number, also a contact number and contact email if they required any other insight regarding usage of the actual device. Samples were collected and returned, and then once that kit was returned and received, then the next set kit was sent to the home address. So here's an overview of participant demographics, pretty self-explanatory, but I think you can quickly see that comparing both the in-clinic and the at-home in terms of the ratio of male-female, fairly equivalent. Again, as I mentioned earlier, as we were monitoring this over the course of enrollment, we did work to even out across male and female by tweaking our ads and trying to appeal more, I guess, in more appealing colors to men. And that being blue. And then age range too across both the groups was fairly comparable. So looking at like early 30s to mid 50s between at home and in clinic. And then the same holds true for the distance from the clinic. So roughly, 5 to just 10 and maybe slightly over 10 miles for the people that were in the at home portion. And now I will turn things over to Mike Walker to carry you forward with our statistical analysis.
Thank you, Robyn. So an interim analysis was pre-specified in the protocol and the statistical analysis plan. It was an interim analysis for early stopping for efficacy. That was conducted. The alpha that was allocated for the interim was P of 0.003, and that left an alpha of 0.049 allocated for the final analysis. At the interim analysis, the results were a p-value of 0.045. Because this p-value did not meet the required p of 0.003, the study was not stopped early for efficacy at that time. And the p-value then required for significance at the final analysis is therefore p less than 0.049. And I'm going to go through the individual results of the pre-specified analyses. These are in order that they were specified in the protocol and in the SAP. So that's the method for controlling for multiple hypothesis testing. It's hierarchical testing. The proportion who consented significantly was significantly higher for the at-home group. The total number of participants was 5,128 who visited the website, and half of those were randomly presented with the clinic website. Half of them would be randomly presented with the home sampling website. The proportion consenting, we had 205 participants who consented, and the table below shows the results. We have the two rows for in-clinic and at-home. The left side shows the consented. The number in clinic was 86 or 3.4%, and the consented at home was 119 or 4.6%. So a larger proportion consented at home than did in clinic, and that difference was statistically significant, p at 0.019. The right-hand side is just showing you the not consented numbers. This is the proportion returning the first, second, and third samples. And again, it was significantly higher for the at-home group. On the table, you can see the rows. The first sample, we have in-clinic and at-home. And below that, second sample in-clinic and at-home, third sample in-clinic and at-home. We'll start with the first sample in-clinic. The number that returned was 36. For the at-home group, the number returned was 57. So the percent returned in clinic was 1.4%. The percent returned from at-home was 2.2%. And, excuse me, the p-value for that was significant, 0.0344. You can see similar results for the second sample and for the third sample. For the second sample, the percent returned was 1.2% for the clinic, 1.8% at home. Again, statistically significant difference, 0.041. And for the third sample, 1% returned for the in-clinic group, 1.5% returned at home, p-value of 0.0345. So for that third sample, a 50% greater return rate, although not surprisingly, overall actual numbers returned are more restricted. We're next going to look at the time to collect an equivalent number of consenting, and we'll see that that was quicker for the at-home group. The Kaplan-Meier, excuse me a second. The Kaplan-Meier plot on the right shows the days to consent for the two groups. In red is the clinic group, in black is the home group, and you can see that the proportion who are not consented, which is the y-axis, the proportion who are not consented is higher in the clinic group. The proportion not consented is lower in the home group, meaning more of them have completed their consent at any given time point. So there were a total of 86 participants who consented in the clinic group. We compared those to the first 86 who consented in the at-home group. And the plot indicates that there is an advantage to the home group. The test for a difference is significant, p less than 0.0001. This was by Cox proportional hazards test. We also evaluated the assumption of Cox proportional hazards, and that is not violated, p-value of 0.017. So it means the effect is consistent across time. We look at the time to return an equivalent number of the first samples, and the Kaplan-Meier plot on the right, again, excuse me, we have the home group in black and we have the clinic group in red. And what we see is over the first interval out to about 20 or so days, the proportion returning the sample was very similar. The two curves are largely overlapping, and then they start to separate out after about 20 days. And what we're seeing is that the home group continues to submit their samples, whereas the clinic group starts falling off. So the plot suggests that there may be an advantage to the home group, but the test for a significant difference gives us a p-value of 0.01, Cox proportional hazards test. In addition, the Cox proportional hazards assumption is violated. It's got a p less than 0.01, and it's very clear when you look at the plot why it's violated, because up to the first 20 days or so, there's really no difference between the two groups in the rate of return. And then that changes. After about 20 days, the two groups are pretty clearly separating. Because of the difference here, it suggests that the hazard ratio is very similar for the first 20 days, but quite different subsequently, and favoring the home group. Because the Cox proportional hazards assumption was violated, we did look at alternative tests, such as the Wilcoxon rank sum test and restricted mean survival time. They gave similar non-significant results, not surprising when you look at the Kaplan-Meier plot. Same analysis for the time to return an equivalent number of second samples, and essentially the same results looking at the Kaplan-Meier plot, and pretty much the same conclusions, which is over the first time interval out to about 30 or so days, the rate of return is very similar in the two groups, but after that they start to separate. But the separation for the overall curve is not significant. That gives us a p-value of 0.3. And because of the violation of the Cox assumption, we again ran the Wilcoxon rank sum and restricted mean survival times, and they remain non-significant. Not surprising given the shape of the curves and the relatively small sample size of 34 in each group. The third sample is very, very similar. Again, we're seeing the curves largely overlapping until they start to separate out later in the study, where for the clinic group, there could be very considerable delays by the time they eventually returned a third sample amongst those who did return the third sample. And the result is not significant, p-value of 0.8. We're going to have a question period at the end, so I'll carry on, but we will have a question period for those of you who would like to ask questions on any of the slides so far. And I think that's back to Neil.
Thank you, Mike. So I'm going to take you through some of the responses to the questionnaires that were completed by both in-clinic and at-home groups after the third sample was collected. So this one demonstrates, so what we've got on the left-hand side is the in-clinic group, and on the right-hand side is the at-home group. So when the in-clinic group were asked whether, instead of traveling to a facility for blood sampling, I would prefer to take the blood sample myself at home and send the sample by mail to the laboratory using the same sampling technology, you can see that a lot of these people that have been going to the clinic to have their samples collected strongly agreed. So we've got approaching 40% of them strongly agreeing that they'd like to collect the sample at home rather than what they'd had to do, and that is going to the clinic. When we reverse that question, so we ask the reverse question to the at-home group, so asking them not would they like to be sampled at home, but would they prefer to be sampled at the clinic? And you can see there we've got over 65% of them strongly disagreeing with that. So the people that had collected samples at home really didn't want to go into the clinic. So that's quite a strong indication of a preference in both groups for home sampling and against in-clinic sampling. Move on to another question. In this case, we've got both the clinic and the at-home sampling on the same graph, the clinic being the orange color, the at-home being the black color. And on the left-hand side, you've got the question, if I took part in a study for new medicine, I would be happy to donate blood sample at a nearby clinical facility with the following frequency. So the frequencies we asked them about was 1 to 2 times per day, once per week, once per month, and would not be willing. And as you can see on the left-hand side, if they were asked to go to a clinical group, there was no real difference between the in-clinic group and the at-home group, and they all had a preference to be sampled between once a week and once a month. Very, very little preference for 1 to 2 times per day, and very little preference for not being willing. However, when we change that question a little bit and ask them whether they'd be willing to donate, how often they'd be willing to donate if that was collected at home, we see for both groups, the in-clinic and the at-home group, a very strong shift to the frequency. So we now see between 40 and over 50% of them being willing to be sampled 1 to 2 times per day rather than once per week or once per month. So that offer of being sampled at home leads to the subjects being willing to be sampled more regularly. So the other questions that we asked are listed out here. I'll just go quickly through them. I'm not showing you the data. The data will be included in the final publication and is in the slide set at the end if anyone wants to take a look at it. But the reason we're not sharing this data is there was no notable difference between the in-clinic and at-home groups. But the other questions we asked were, before participation, I was given all the information I needed in relation to the study, so no difference between the two groups there. Before participation in this study, I did not have any problems with giving blood samples using standard venipuncture approaches, so we'd not selected any differences in the groups there. Donating the blood sample has not interfered with my daily schedule. That was agreed by both groups. Donating the blood sample was not painful. No difference in the two groups, and you can show the data, but it wasn't found to be a painful experience. I had a good experience from taking part in the study. No difference between the two groups there, and I would recommend participation in similar studies to others. No difference between the two groups. So something that was obviously also of interest to us is what is the quality of the samples that are collected. And this has been assessed. The data that's shown here, this was assessed by both the clinic facility and the analytical facility. What we're showing here is the data from the analytical facility at the University of Ghent in Belgium. And what you can see at the top, the top 3 pie charts are the at-home group, and the bottom one is the in-clinic group. And this has been able to fill at least one tip, so therefore being able to get a valid result. And you can see for the at-home, to begin with, they start off at 72%, were able to successfully get a sample, whereas the in-clinic was in the high to mid-80s. So far more success in the in-clinic group, which is what we would expect, I guess, because these samples are being collected by a healthcare professional in the clinical setting versus self-collection at home. But what we can see, particularly in the at-home group, is once we move to sample 2 and sample 3, we get an increase in success rate. And so as people got used to the device, they increased their success of collection. And we saw a very similar trend for people collecting at least 2 sub-aliquots or all 4 sub-aliquots being successfully collected for each sample. So just to conclude on our findings from this study, as you saw, the proportion consented was significantly higher for the at-home group. The proportion returning the first, second, and third samples was significantly higher for the at-home group. The time for equivalent number of consenting was quicker for the at-home group. And there's a suggestion there's a shorter period between consent and return of the first, second, and third samples for the at-home group. But those findings were not statistically significant. We saw clearly from the questionnaires that patients are happier to collect samples at home rather than travel to a facility. And we saw that at-home sampling is likely to be more feasible for frequent sample collections. We saw the success rate for at-home collection was lower than the in-clinic group, but that improves with successive sample collection for the at-home group. And we very much recognized the need for potentially improved training and support, particularly of the at-home group. And I think a lot of people running these sort of studies have learned a lot more about that since we got this protocol approved. So no further ado, I think that's the last. Oh yes, just want to go on to some thank yous before the Q&A. So this study only ran because a lot of people got involved and were willing to give their time and resources for free. So you can see a very large list of people from a variety of different organizations who contributed their time and resources to writing the protocol, the recruitment, the analysis of the samples, the analysis of the data, and the reporting. We also did need some money to come in to pay for advertising and some of the activities we got. So we thank George AT, IQVIA, Roche, Tasso, and Velocity for their donations that meant the financial parts of this study were able to go ahead. So with no further ado, I'd like to take questions on this. I will say before we start the questions that the team are currently starting the process of writing a manuscript on this data and taking it to a number of international conferences in the coming months. But we're very excited to see what questions you have. So let's come in and see what we've got in here. So we've got from Sandeep. Thank you for your question, Sandeep. Will you conduct a follow-on research extended study with a higher N population than 31? So Sandeep, I think, if other people want to chip in on this, it took us 2 years to recruit the number of people we had. We simply don't have the resources to, as PCSIG, to conduct a follow-on study. The kind of advertising and recruitment that pharmaceutical companies can do is out of our reach. So it'd be great if someone was able to do a study. But the fact is, even with the numbers we got, most of our outcomes were statistically valid. I don't know, Robyn and Mike, do you want to come back on stage and answer any of the questions?
Well, there were over 5,000 participants who visited the website, and for the various outcomes that we were interested in, it ranged up to a few hundred for whom we actually had samples or the required data. I think one of the key lessons is that it's actually easier to get participants using the at-home collection method than asking people to come into clinic. And I think that's one of the things that should be considered in other clinical trials.
Yes. Any comments on Sandeep's question at all, Robyn?
I mean, I agree with you and your point, Neil, in terms of the duration that it took for the recruiting, and just the cost implications. I mean, I think for our group to move forward, right, with more of an expanded study would just not be feasible.
Yeah.
Yeah.
Question from Eric. For the at-home collection, were the patients able to ship their samples from their home, or did they have to drop them at a post office, UPS, etc.? And I believe the answer is they were shipped back to the same site as the clinic, and they were able to ship them directly from their home, is my belief. Is that correct, Robyn?
I believe so, yes. Yep.
Question from Haley. Were in-clinic participants provided with money to cover their travel and expenses? From memory, I don't think they were they, Robyn?
No, nothing other than the stipend, which was the per collection. They received a $10 fee, but no travel sort of costs were covered. Yeah, and if we recall from just the distance, I mean, it was a reasonable distance from the clinic. So, no more than 10 miles or so.
Yes.
Yeah.
So question from Brian, were the home subjects in an urban setting, a rural setting, or both? So the clinic was in an urban setting. And as I think, I don't know whether you want to answer this one, Robyn.
I don't know specifically like how you would classify the addresses of the participants.
The distances from the clinic were maybe a little further than the in-clinic people, but they were all within, all less than 40 miles, really.
Right, exactly. Yeah.
So, but we can that is something maybe we could take away, Brian, and have a little bit more of a look at that, if there's any data we can get out of that to put out there in the publication. I tried to do a geographic map of their postcodes in Excel, but couldn't make it work. So maybe we'll have a little bit more of a go at that and see if it gives us any trends. Maria, were you able to monitor whether the at-home participants utilize the support email, phone numbers, additional services? I guess that number is monitored. We don't have it to hand, unless you know anything else, Mike and Robyn.
No,
I do not. Sorry.
no. Mm-mm.
But yeah, it is recorded, Maria, and maybe that's something we can incorporate into the publication as well. It's a really good point. Thank you. Question from Steve. Interesting that at-home sampling, even with instructions, benefited from practical use of the Tasso device. Presumably this suggests doing some sort of dummy collection before start of study would be recommended. Anyone want to have a go at that one?
You mean in terms of almost like training, if you will?
I think that's what they mean.
Yeah.
Practice.
I mean, I think there's advantage to that. And then also even more explicit instruction, maybe like a video of some sort that could be sent electronically, demonstrating usage. Because there was some inconsistency, right, in terms of the quality of the sample. And it could certainly be related to that, right?
I know a lot of people that are using this for clinical trials in healthcare. Since we wrote our protocol and designed the way we were sampling, have found that if the first sample for the at-home group is collected in a clinical setting with that kind of training, then they get much higher success rates for all the subsequent sample collections. So a lot of people kind of recommend, and you can imagine for a routine pharmaceutical clinical trial, if on their first visit they're able to take a blood sample under supervision in the clinic, hopefully that would improve the response, the quality of the samples when they go home. But we simply weren't able to do that with this study design. Any other comments around that, Robyn or Mike?
I mean, I think it's definitely a valid point to consider for sure.
Agree.
So Katie, what were the main drivers for lower quality in home collected samples? Was it volume, hemolysis, etc.? So let's see. It's actually written in there. I just need. Let's have a look on a bigger screen. So in here, the orange is overfilled, and the grey is empty, and the red is underfilled. So the majority were underfilled that failed rather than overfilled or no sample at all. So the majority of the failures were red. Does that? So nothing to do with hemolysis. These were, just so everyone knows, these were dried samples because we knew our recruitment period was going to be long and all the samples were going to get analysed together for cost benefit reasons. So we needed samples that could be shipped and stored for a long time, and so they're all dried blood samples. So hemolysis was not assessed. Lewis, thanks Robyn, Michael and Neil for a valuable presentation. Were there any adverse device effects, device deficiencies and/or complaints during the study? Not that I'm aware of.
No, I don't think so. No.
There was one adverse event reported. It was something pretty minor. I'm not remembering exactly what it was, but I was quite surprised actually that there was literally only one reported adverse event in the whole study.
So any more? I'm not seeing any more questions come in at all. But please, we're happy to, if you have any questions after the event, please, just send them in. And as I say, the plan is to publish this. Any other comments at all, Robyn, Mike?
No, I don't believe so.
Nope.
No. Yeah, I mean, we look forward to carrying this message.
Yeah, absolutely.
Ahead, yeah, absolutely. Yeah, definitely.
So late comment by Steve, seems underfilling was the biggest issue that didn't change. And yes, that seemed to be the case, but was a much bigger issue with the at-home than the in-clinic.
There really was fairly minimal training and instruction. Retrospectively, it would have been good if we'd had them come in and do formal training and do a dummy collection. And, I think going forward in other studies, those are certainly things that we would want to do to try and improve the quality. And I would expect it to improve.
Absolutely. And there's been a number of publications of data showing high, if that kind of training is done, then the quality of the samples is improved. Okay, well, I'm going to just talk a little bit about PCSIG and upcoming events. If anyone has any questions that pop into their mind while I'm going through that, please type them in the chat. Happy to still cover them. We've got plenty of time. So our next webinar coming up on May the 21st is biomarkers of colorectal polyps from at-home collected dry blood spots. So that's Ben Herbert, the Chief Scientific Officer of Sanguine Bio in Australia. So poor Ben's going to be up in the middle of the night Australia, his time, to be able to meet the criteria for Europe and North America. So thank you, Ben, for being willing to do that. And, please take a look at that and register for that event. We have monthly events, as I say, booked out for about over a year into the future. So there'll be plenty of content, and all our existing content is available on YouTube. Just a reminder, if you don't know about Patient-Centric Sampling Interest Group, we're a not-for-profit group looking to bring together different organizations who are passionate about bringing forward patient-centric sampling, increasing understanding, increasing adoption where it's appropriate. If you want to visit our website, the QR code on the left should work. If you want to join our LinkedIn group, which is free, please use the QR code on the right. And in order to make all these webinars free and all the activities that we have for our members free, we're very much reliant on our sponsors, and we thank them very, very much for contributing towards our organization. So that's all I had. I'll just check whether there's any more questions. I don't believe there are.
So no, yeah.
So thank you, everyone. Thank you for your attendance, and this webinar has been exciting, and we'll be publishing it quite soon. Bye.
Thank you, everyone.